Welcome to our video on everything about neurons. Right now, inside your prefrontal cortex, individual pyramidal cells are executing non-linear computations that render our most advanced silicon architectures primitive by comparison. We aren't just looking at simple biological switches, we're dissecting the high-stakes bioenergetics of the sodium-potassium pump, a molecular engine consuming 20% of your total daily calories just to maintain resting potential. Beyond basic anatomy lies the deep lore of dendritic spikes and the precise geometry of spines that physically rewire in seconds. We're bypassing the textbook basics to explore the specific capacitance of the lipid bilayer, the legacy of the Golgi-Kajal feud, and the mystery of ephactic coupling, where neurons communicate through localized electromagnetic fields without even firing a synapse.
This field effect signaling is only the preamble to the intricate machinery of the snare complex, a protein-driven docking bay where synaptic vesicles are primed for fusion in less than a millisecond. We have to address the war of the soups and the sparks, the decades-long ideological battle between Sir Henry Dale and John Eccles over whether neural transmission was fundamentally chemical or electrical. While Dale's soups eventually won the primary narrative, the discovery of gap junctions via CaMKII proteins proved the sparks were right all along in specific high-speed circuits. Look at the nodes of Ranvier, where sodium channel density hits a staggering 1200 per square micrometer. This isn't just insulation, it's a strategic optimization of the lambda length constant, ensuring the action potential regenerates with lossless fidelity across the axonal cable.
But the fidelity of the axonal cable is only half the story. To understand how a neuron actually learns, we must track backpropagating action potentials. These voltage spikes defy standard directional flow, surging from the axon hillock back into the dendritic arbor to signal the entire cell that it has successfully fired. This backward wave is the physical trigger for spike timing dependent plasticity, where a precision window of just 10 milliseconds determines whether a synapse is reinforced via AMPA receptor trafficking. Beneath this electrical storm, the axon's interior is a logistical masterpiece managed by kinesin 1 motor proteins. These molecular machines walk along microtubule tracks, hauling cargo at 400 millimeters per day through a cytoplasm that behaves like a high-viscosity gel. We even see neurons breaking directionality via retrograde signaling, releasing endocannabinoids like 2-AG to travel backward across the cleft and suppress presynaptic release, effectively allowing the receiver to dictate the terms of the message.
This bidirectional control is further complicated by the coincidence detection of the NMDA receptor, which remains functionally silent behind a magnesium ion plug until a specific depolarizing threshold is met, acting as a literal biological logic gate. To model this, we look to Wilfrid Rall's Keeble theory, which transformed our understanding of neurons from simple points into complex cylinders, defined by specific axial resistance and membrane time constants. When an inhibitory shunting synapse opens chloride channels near the soma, it doesn't just subtract voltage, it performs a division operation, effectively zeroing out distal dendritic inputs before they reach the trigger zone. This is further bolstered by the reality of somatic mosaicism, where retrotransposons like line 1 elements physically alter the DNA of individual neurons during neurogenesis, ensuring that even adjacent cells of the same class are genetically distinct processing units.
This genetic heterogeneity is mirrored by the metabolic symbiosis of the tripartite synapse, where astrocytes actively regulate the glutamate-glutamine cycle through EAAT transporters to prevent excitotoxicity. While 20th century dogma asserted a 10-to-1 glia-to-neuron ratio, Susanna Herculano-Huzel's isotropic fractionator research revealed a near 1-to-1 parity in the human brain. Within these circuits, we find specialized architectures like von Economo neurons, featuring an elongated morphology optimized for rapid long-distance integration across the anterior cingulate. Even the axon's interior is a marvel of spatial organization, containing a periodic cytoskeleton of actin rings spaced exactly 190 nanometers apart. This lattice doesn't just provide tension, it scaffolds the proteins that cluster ion channels, ensuring the membrane is tuned for high-frequency bursts while mitigating the energetic cost of heat dissipation during repetitive firing.
This spatial precision extends to the decentralized manufacturing of proteins, where the neuron bypasses the nuclear hub through local translation. Ribosomes are sequestered directly at the base of dendritic spines, allowing for the immediate synthesis of receptors from idling mRNA transported via ribonucleoprotein granules. This is the foundation of the synaptic tag and capture hypothesis, where a stimulated synapse creates a molecular marker that catches these traveling proteins to solidify a memory. Encasing these clusters are perineuronal nets, a glycocalyx lattice that acts as a molecular brake, restricting the lateral diffusion of AMPA receptors to terminate critical periods of neuroplasticity. We only understand these biophysics because of J.Z. Young's 1936 rediscovery of the squid giant axon. At nearly 1 millimeter thick, it provided the volume required to prove that the axoplasm is a highly conductive electrolyte. This environment is further regulated by HCN channels, which generate the funny current, a hyperpolarization-activated inward flow that maintains rhythmic pacemaking and prevents the neuron from falling into a terminal silent state.
This homeostatic balance is anchored by the postsynaptic density, a specialized protein matrix where over 1,000 distinct protein species cluster into a 30 nanometer thick processing hub. At the heart of this architecture is CaMKII, a dodecameric enzyme acting as a molecular hard drive. Its symmetry allows for autophosphorylation, preserving a memory of calcium influx long after the signal dissipates. Beyond existing synapses, the embryonic growth cone utilizes filopodia to sense femtomolar concentrations of netrin and slit guidance cues, steering axons through the brain's white matter with micron-level precision. We are even discovering human-specific cell types like the Rosehip neuron, a GABAergic regulator discovered in 2018 that is entirely absent in rodents, proving our inhibitory architecture underwent a radical evolutionary divergence to manage the extreme computational density and unique circuit motifs of the human neocortex.
This architectural divergence is further nuanced by volume transmission, where neurotransmitters like dopamine and serotonin bypass the synaptic cleft entirely, diffusing through the extracellular matrix to modulate vast neural ensembles via high-affinity extrasynaptic receptors. This wireless signaling is governed by the tortuosity of the brain's interstitial space, creating a chemical gradient that dictates the global state of the circuit. On a molecular level, the opening of a single ion channel is a mechanical feat. The S4 transmembrane segment, acting as a voltage sensor, contains positively charged arginine residues that physically torque in response to membrane potential shifts. This movement generates a measurable gating current, a discovery enabled by the 1970s breakthrough of the patch clamp technique. By establishing a gigome seal with a glass micropipette, Erwin Neher and Bert Sackman were able to record the stochastic flickering of individual proteins for the first time. To sustain this electrical overhead, mitochondria are not merely drifting, they are actively recruited and anchored at active synapses by the Miro and Milton protein complexes, ensuring a localized ATP supply for the grueling work of clathrin-mediated endocytosis during vesicle recycling.
This metabolic demand is matched by the structural rigidity of the axon initial segment, or AIS, a high-density filter scaffolded by the protein anchorin G. This region serves as the ultimate gatekeeper, adjudicating the decision to fire through a hyper-localized concentration of NAS 1.6 channels, which possess a significantly lower activation threshold than the NAV 1.2 variants found in the distal axon. Yet the most jarring aspect of neural evolution is the viral origin of myelin itself. Recent genomic analysis reveals that the RNLTR1 retrotransposon, a remnant of an ancient retroviral infection, is the essential genetic trigger for myelin-basic protein synthesis in jawed vertebrates. Without this prehistoric molecular hijacking, saltatory conduction would be impossible, requiring axons so thick that a functional human brain would physically exceed the volume of the skull just to achieve current processing speeds.
This viral legacy is complemented by the kiss and run fusion mechanism, a lightning-fast alternative to full vesicle collapse, where a 50-nanometer vesicle releases its cargo through a transient pore without losing its structural identity to the plasma membrane. This flicker of connectivity is governed by the VATPH proton pump, which creates a highly acidic internal pH of 5.5 to drive neurotransmitter concentrations up to 100 millimolar, a chemical gradient so steep it requires specialized V-GLUT transporters to prevent leakage. We see further modulation in the synaptic co-release of ionic zinc, which acts as a precision brake by binding to the amino terminal domain of GLU-N2A subunits. This isn't just chemical noise, it's a localized regulatory system that prevents excitotoxic damage during high-frequency bursts. Even the physical architecture of the active zone is choreographed by the CAZ, the cytomatrix at the active zone, a dense scaffolding of piccolo and bassoon proteins that physically tether vesicles in a hexagonal lattice, ensuring they are positioned within nanometers of the voltage-gated calcium channels for near-instantaneous release.
This structural rigidity is mirrored by the emerging physics of liquid-liquid phase separation, where proteins like synapsin-1 condense into membraneless droplets. These droplets sequester pools of vesicles into a high-density liquid phase that remains distinct from the surrounding cytoplasm through sheer molecular density. Deep within the dendritic arbor, the smooth endoplasmic reticulum functions as a neuron within a neuron, a continuous intracellular highway for calcium signaling that can propagate waves of depolarization independent of the plasma membrane. This system is fueled by the astrocyte neuron lactate shuttle, where glia convert glucose into lactate for export via MCT2 transporters, ensuring metabolic continuity during intense firing. To observe the extreme limits of this throughput, we look to the calyx of Held, a gargantuan synapse where a single presynaptic terminal utilizes over 500 active zones to achieve a temporal precision measured in the low microseconds.
This microsecond precision is underpinned by the stochastic nature of quantal release, a phenomenon first quantified by Bernard Katz and Paul Fatt. They discovered minis, spontaneous sub-threshold voltage blips occurring without an action potential, proving that neurotransmitters are released in discrete fixed-sized packets rather than a continuous stream. Moving to the population level, we encounter the paradox of sparse coding. The vast majority of neurons remain functionally silent at any given moment, with fewer than 1% of the population firing to represent a specific stimulus to prevent metabolic collapse. This network is actively sculpted by microglia, which utilize the C1q and C3 complement cascade to physically ingest and prune weak synapses during sleep. This remodeling is synchronized with the glymphatic system's pulse-driven convection, where AQP4 water channels on astrocytic end feet facilitate the clearance of interstitial solutes, effectively resetting the neural environment for the next processing cycle.
Beyond this clearance, the structural integrity of the synapse is maintained by the transsynaptic nanocolumn, where neurexin neuroligand bridges align release sites with receptors with 80 nanometer precision. This spatial coupling is ruthlessly governed by chandelier cells, specialized interneurons whose axonal partridges terminate exclusively on the axon initial segment. This inhibitory dominance is predicated on the KCC2 transporter, which maintains the low intracellular chloride concentration necessary for hyperpolarization. If KCC2 expression is compromised, the chloride reversal potential shifts, causing GABA to paradoxically depolarize the neuron, a biochemical flip that drives pharmacologically resistant epilepsy. This localized control is supported by Golgi outposts, satellite organelles located in distal dendrites, which allow the neuron to process and traffic complex membrane proteins locally without relying on the slow centralized transport from the primary cell body.
This localized autonomy extends to the mechanical pulse, a controversial frontier where the soliton model suggests action potentials are not just electrical, but thermodynamic waves of adiabatic pressure.
As the voltage spike travels, the lipid bilayer undergoes a reversible phase transition from fluid to gel, physically expanding the membrane by several nanometers. This mechanical displacement is sensed by piezo-1 channels, which translate the cell's physical distortion into an immediate influx of calcium. This biophysical feedback is further refined by the subthreshold resonance of the membrane's impedance profile. By precisely tuning the density of M-type potassium channels, a neuron can act as a biological bandpass filter, selectively amplifying inputs at specific frequencies while ignoring asynchronous noise. This frequency preference is the hidden oscillator that allows individual cells to resonate with the massive, coherent theta rhythms of the hippocampus, transforming the neuron into a frequency-dependent signal processor.
This resonant filtering is further refined by the primary cilium, a solitary organelle protruding from the soma that serves as a specialized signaling hub for the sonic hedgehog pathway. Once dismissed as vestigial, the cilium's restricted volume concentrates GPCRs to detect morphogenic gradients with extreme sensitivity. This sensing is matched by the unique biophysics of T-type calcium channels, which operate via a deactivation mechanism. They require a brief period of hyperpolarization before they can trigger a low-threshold spike. This rebound burst firing is the primary driver of rhythmic thalamic oscillations. Protecting these high-activity zones is neuromelanin, a dark polymer that acts as a high-capacity sink for redox-active iron and toxic catechols. By sequestering these reactive species, neuromelanin prevents the oxidative degradation of the substantia nigra, allowing these specialized dopaminergic neurons to survive the intense metabolic stress of a human lifespan. This mechanical protection is mirrored by the link complex, a molecular bridge that physically tethers the nucleus to the surrounding cytoskeleton. This connection allows the neuron to perform direct mechanotransduction, where physical tension on a dendrite can instantly pull on the nuclear envelope to trigger the remodeling of topologically associating domains. Within these three-dimensional DNA folds, the neuron sequestered an immense library of circular RNAs. These circRNAs act as molecular sponges, titrating the levels of microRNAs to provide a stable long-term regulatory buffer that can persist for weeks, far outlasting standard linear transcripts. This stability is essential for maintaining the high-fidelity information throughput measured by the Landauer principle, which defines the absolute minimum energy required to erase a single bit of neural information. To push these physical limits, specialized pacemaker neurons in more mirrored electric fish have evolved to fire with a temporal jitter of less than 1 microsecond, operating at the extreme boundary of deterministic biological signaling, where even the thermal noise of individual water molecules must be filtered to prevent bit flip errors in the neural code.
This thermodynamic stability is further challenged by the hydration shell of the ion channel itself, where exactly 6 water molecules must be stripped from a potassium ion before it can pass through the selectivity filter's carbonyl oxygen rings. This sub-angstrom coordination is so precise that substituting deuterium increases solvent viscosity, measurably slowing the gating kinetics. In the primary motor cortex, this throughput is pushed to the extreme by Betz cells, massive pyramidal neurons with cell bodies exceeding 100 micrometers. To maintain these giants, the neuron employs MHC class I molecules, not for immune defense, but as signaling hubs that recruit the PIRB receptor to prune excess synapses. This logistical overhead is managed by the dynin-dynactin complex, a -N-directed motor hauling cargo back to the soma at 1 micrometer per second, preventing distal axonal congestion.
This retrograde transport is the biological prerequisite for the signaling endosome, a specialized vesicle that carries nerve growth factor all the way to the nucleus to prevent the activation of the caspase-3 apoptotic pathway. While survival is managed at the soma, the neuron's true computational power is hidden in the information-theoretic capacity of its synapses. Recent Bayesian modeling of the hippocampal CA1 pyramidal cell suggests that each synapse can store approximately 4.7 bits of information, categorizing synaptic strength into 26 distinct discrete levels rather than a simple binary state. This granularity is refined during the postnatal GLU-N2B to 2A subunit switch, a developmental milestone that truncates the decay time of NMDA currents from 300 milliseconds down to 50, drastically increasing the temporal resolution of the circuit.
This chemical specificity honors the legacy of Otto Loewy's 1921 Vega stuff discovery, where he proved chemical transmission by transferring the perfusate from a stimulated frog heart to a denervated one, identifying the first neurotransmitter, acetylcholine. We now know this diffusion is physically constrained by the glycocalyx, a 10-nanometer thick carbohydrate forest that exerts entropic forces to prevent receptor escape, ensuring that the molecular signal remains localized within the high-fidelity zone of the postsynaptic density.
This chemical landscape is further nuanced by the discovery that D-serine, rather than glycine, is the primary endogenous coagonist required for NMDA receptor activation in the telencephalon. Synthesized by serine racemase, D-serine levels dictate the sliding modification threshold of the Bienenstock-Cooper-Munro theory, ensuring the tripping point for synaptic plasticity scales with the neuron's history of activity. To physically accommodate these shifts, the neuron secretes matrix metalloproteinase 9, an enzyme that locally proteolyzes the extracellular matrix to facilitate spine remodeling, transitioning our understanding to a tetrapartite synapse. This structural fluidity is checked by the extreme longevity of nucleoporins. Unlike almost every other protein, these age-invariant scaffolds are rarely replaced, making them the primary site where oxidative damage eventually compromises the neuron's transcriptional gatekeeping over a human lifespan.
This transcriptional decay is the ultimate bottleneck, but the mathematical foundation of the action potential itself was forged through even more grueling labor. In 1952, Alan Hodgkin and Andrew Huxley solved the nonlinear differential equations of neural excitability using a Brunsviga mechanical hand-cranked calculator, a feat of manual iteration that defined the M, N, and H gating variables. These kinetics are modulated by the CaMKII encoded M current, a unique potassium flow that never fully inactivates. By maintaining a constant leak at sub-threshold voltages, the M channel acts as a programmable resistor that prevents stray synaptic noise from accidentally triggering a 100 millivolt spike. This is further refined by the secret flexibility of the all-or-none law. At the nanoscale, the action potential's amplitude fluctuates based on the local concentration of extracellular divalent cations like magnesium, which screen the membrane surface charge to shift the voltage sensing threshold in real time.
This ionic screening is mirrored by the precise geometry of the synaptic cleft, which was only definitively visualized in 1954. Sanford Pillay and George Palade utilized the first high-resolution electron micrographs to reveal the 20-nanometer gap, finally providing the smoking gun that killed the reticular theory. Within this gap, the local concentration of chondroitin sulfate proteoglycans creates a polyanionic environment that traps cations, effectively acting as a biological capacitor. To fuel this, the neuron engages in a subtle thermal cycle. A single action potential generates a transient temperature spike of precisely 2 microkelvins. This heat is almost entirely reabsorbed during the repolarization phase, characterizing the nerve impulse as a nearly adiabatic process. This thermodynamic efficiency is even linked to biophotonic signaling, where neurons emit ultra-weak bioluminescence during oxidative stress. Emerging models suggest that the high refractive index of the myelin sheath may allow it to function as a biological fiber optic cable, facilitating sub-nanosecond photonic communication that bypasses the sluggish limitations of ionic diffusion.
This high-speed logic is mirrored by the persistent sodium current, or INAP, a kinetic state where sodium channels fail to fully inactivate, generating a steady-state inward flux that perpetually nudges the membrane towards threshold. This simmering voltage transforms the neuron into a frequency-dependent resonator. This tuning is further refined by A-type potassium currents, which utilize Schall and Shaker protein families to act as transient shock absorbers, delaying spikes to filter out asynchronous noise. This high-stakes gating is governed by the safety factor of transmission, the ratio between available synaptic current and the firing threshold. Within the nucleus, we find A to I RNA editing mediated by ADAR enzymes, which physically recodes glutamate receptor transcripts to swap glutamine for arginine, a single atom change that renders the channel impermeable to calcium and prevents metabolic collapse.
This enzymatic recoding is a localized defense against the failure of the QR site, but the neuron's true resilience resides in the buffer-limited diffusion of its calcium microdomains. Within these 10 nanometer zones, calcium concentrations spike to 100 micromolar in microseconds, strictly localized by parvalbumin, a protein acting as a kinetic sponge to prevent signal crosstalk. This precision echoes the negative variation observed by Émile Dubois-Reymond in 1848, which first proved the nerve impulse was a quantifiable reduction in a resting current. This leads to the synaptic ribbon, a specialized organelle in retinal bipolar cells that uses a proteinaceous conveyor belt to maintain continuous glutamate release at rates exceeding thousands of vesicles per second. This throughput is governed by the Henneman size principle, a recruitment logic ensuring the smallest motor neurons fire first to facilitate smooth, graded force production.
This recruitment is physically throttled by the axon cap of the teleost-Mauthner cell, a specialized neuropile where a localized extracellular inhibitory potential silences the trigger zone through pure field effect interference. This biophysical leverage is maintained by the NAT calcium exchanger, which operates at a strict 3:1 stoichiometry, leveraging the sodium gradient to extrude calcium against a 10,000-fold concentration steepness. Beneath this ionic tension, we find silent synapses, glutamate-blind connections populated exclusively by NMDA receptors. These latent nodes are only recruited through the rapid insertion of GluA2-lacking AMPA receptors, which are uniquely sensitive to polyamine block, creating an inward rectifying current that acts as a developmental gate. This matures alongside the hippocampal mossy fiber detonator synapses, where massive presynaptic terminals utilize filopodial extensions to target inhibitory interneurons, allowing a single granule cell to exert a disproportionate veto over the entire CA3 network through feed-forward inhibition.
This feed-forward veto is even more extreme in the cerebellar pinceau, a specialized structure where basket cell axons wrap around the Purkinje cell's axon initial segment like a terminal fringe. Here, inhibition is achieved through a hyper-ephaptic field effect, where the basket cell generates an extracellular voltage drop so sharp it physically pulls current away from the Purkinje cell, preventing spike initiation without any neurotransmitter release. This architecture is scaffolded by Neurofacin 186, a cell adhesion molecule that locks the inhibitory terminal in place. To sustain the resulting kilohertz range firing, these circuits rely on BK channels, calcium-activated potassium pores with a staggering 250 picosiemen conductance. These big potassium channels act as the ultimate repolarization engine, while the underlying vesicle logistics consume over 11,000 ATP molecules per second just to maintain a primed state, proving that neural silence is an active, high-energy investment.
This high-energy investment is mathematically optimized by the G ratio, the fundamental quotient of the inner axonal diameter to the total fiber diameter. Evolution has converged on a value of exactly 0.6, the precise geometric sweet spot where conduction velocity is maximized without exceeding the metabolic limit of myelin volume. Within this architecture, the neuron employs nitric oxide as a gaseous retrograde messenger that ignores the lipid bilayer entirely, diffusing across 20 micrometers to activate soluble guanylyl cyclase in the presynaptic terminal. This chemical flux is structurally stabilized by the Schenck-Homer scaffold, a massive protein lattice that physically breaches the plasma membrane to the intracellular calcium stores of the endoplasmic reticulum. This coupling allows the neuron to maintain self-organized criticality, a state where branching ratios hover at exactly 1, ensuring that signal propagation remains stable without collapsing into total neural silence or escalating into a runaway seizure.
To navigate this critical state, neurons rely on a molecular barcode of clustered protocaderons, a combinatorial system where alternative splicing generates over 50,000 unique protein identities. This facilitates a rigorous mechanism of self-avoidance, ensuring a neuron's expanding dendritic arbor recognizes its own branches to prevent redundant self-synapsing. This connectivity is further refined by tenurins, ancient proteins that pair with latrophilin receptors to form a high-affinity molecular velcro, dictating target cell specificity during circuit assembly.
This spatial sorting is enforced by TRIM46 and nucleoporin 153, which reside at the axonal entry point to act as a physical sieve, selectively excluding dendritic organelles from the unidirectional axonal highway. Beneath this architecture, the sigma-1 receptor at the ER mitochondria interface acts as a metabolic rheostat, titrating calcium flux to prevent autophagic collapse during the extreme bioenergetic demands of sustained computational loads.
This metabolic equilibrium is defended by the massive hypertrophy of the neuronal nucleolus, which produces a constant stream of ribosomal RNA to support a proteome that must be refreshed every few days. Neurons utilize a unique microexon splicing program, where segments of DNA as short as 3 base pairs are inserted into proteins to radically alter their binding affinity for synaptic scaffolds. This hyperspecific splicing is regulated by the protein SRRM4, ensuring the same gene produces different functional isoforms across cell classes. This molecular diversity is the precursor to the dark neuron phenomenon, a state where cells undergo a rapid reversible contraction and cytoplasmic densification. While often dismissed as a fixation artifact, evidence suggests this shrunken state is a protective low-energy mode triggered by transient metabolic stress. This is further supported by pioneer axons, which physically pave a trail of tag-1 adhesion molecules to create a structural highway. These paths are maintained by the glycan shield, a dense coat of polysialic acid on NCAM proteins that reduces steric hindrance, allowing axons to slide past one another in high-density bundles without triggering premature mechanical stalling.
This guidance is dictated by Myelin, a massive glycoprotein secreted by transient Cajal-Retzia cells to orchestrate the inside-out radiomigration of the neocortex. By activating VLDLR receptors, Myelin triggers the phosphorylation of the adapter protein disabled 1, acting as a molecular stop signal to prevent migrating neurons from overshooting their designated cortical lamina. This laminar precision is mirrored in the mRNA architecture of the postsynaptic density, where G-quadruplex structures, 4-stranded helical knots, gate the translation of PSD95 to prevent premature synaptic strengthening. This molecular gating is essential in parvalbumin-positive interneurons, where the specific kinetics of NAV 1.1 channels allow for the 100 Hz firing rates that underpin gamma-band synchrony. A single missense mutation in the SCN1A gene doesn't just weaken inhibition, it desynchronizes the entire cortical ensemble, turning a sub-nanometer shift in a voltage sensor into the catastrophic network collapse of Dravet syndrome.
This sensitivity to voltage-gated kinetics is mirrored by the structural plasticity of the axon initial segment itself, which isn't a fixed landmark. In response to chronic overstimulation, the entire AIS can physically migrate up to 10 micrometers distal from the soma. This relocation, regulated by the calcium-dependent phosphatase calcineurin, increases the neuron's input resistance to prevent excitotoxic burnout. This structural shift is fueled by the glycogen shunt in astrocytes, where glycogen is converted to lactate and shuttled through MC2 transporters to meet the localized ATP demands of axonal remodeling. We see a similar mechanical precision in the clinging phase of neurogenesis, where the 40 kilodalton protein double-cortin stabilizes microtubule tracts. Without this stabilizer, the radioglia scaffold collapses, leading to lissencephaly, proving that a neuron's final identity is a direct consequence of its mechanical velocity during embryonic development.
This developmental velocity is sustained by the Albers post-cycle of the sodium-potassium ATPase, a high-frequency conformational oscillation where 3 sodium ions are occluded within the E1p phosphorylated state before the enzyme flips to the E2p configuration. This molecular inversion defines the kinetic limit of the neuron's recovery phase after a high-frequency burst. To prevent network saturation, neurons execute homeostatic scaling via the immediate early gene Homer1A. During slow-wave sleep, Homer1A surges into the dendritic spine to competitively displace long-form Homer proteins, effectively uncoupling the scaffold to downregulate synaptic weights globally. This synaptic scaling is further refined by pan-Axon-1 hemichannels. Unlike gap junctions, these large pore channels act as high conductance valves for ATP release, activating P2X7 purinergic receptors to create a pyranome signaling layer that adjusts the gain of the local circuit independently of traditional synaptic release.
This extracellular modulation is mirrored by the geometric rigidity of the paranodal junction, where the 155 kilodalton isoform of neurofacin interlocks with the Casper-contactin complex. This molecular zipper creates a high-resistance barrier that prevents ion leakage from the node into the periaxonal space, a gap of only 20 nanometers, where the membrane potential is shielded from the bulk extracellular fluid. Within this microenvironment, the neuron employs carbonic anhydrase 14 to tether pH fluctuations, ensuring that the rapid extrusion of protons doesn't prematurely desensitize acid-sensing ion channels. This pH buffering is critical for the high-fidelity signaling of the MAP2 stabilized cytoskeleton within the dendritic shaft. Here, the selective transport of poly-A binding proteins ensures that while the soma remains the genetic hub, the distal dendrite maintains its own proteomic state through the persistent localized translation of ARC and PKM-zeta, the atypical protein kinase responsible for the perpetual maintenance of long-term potentiation. This persistence is countered by the resurgent sodium current, a biophysical anomaly where the NaV 1.6 channel pore is temporarily obstructed by an intracellular blocking peptide during repolarization. This prevents terminal inactivation, enabling specialized cells in the auditory brainstem to fire at 500 Hz without refractory failure. Supporting this throughput is the molecular scaffolding of the Cast and Elks proteins, which exert a physical tension holding the re-releasable pool of vesicles at a standoff distance of precisely 3 nanometers from the membrane. This spatial rigging is protected by the chaperone CSP alpha, preventing snare misfolding during the thermal stress of high-frequency bursts. On a circuit level, this precision facilitates anti-Hevian plasticity in the dorsal cochlear nucleus, where synapses weaken during coincident firing to subtract internal biological noise, like the sound of your own heartbeat, from the primary auditory stream.
This noise subtraction is further refined by the axon-carrying dendrite morphology, a structural anomaly where the axon originates directly from a primary dendrite rather than the soma. In these high-performance interneurons, synaptic inputs bypass the somatic filter, allowing distal signals to trigger spikes with zero latency integration. This unconventional geometry is complemented by the non-conducting functions of Kv 2.1 channels. Instead of just passing ions, these proteins cluster into massive micropatches of up to 100 channels that anchor the endoplasmic reticulum, creating specialized calcium signaling couplons. This rigidity is countered by the synaptic triad in the olfactoid glomerulus, where a single presynaptic terminal simultaneously excites a mitral cell and an inhibitory periglomerular cell. This signal utilizes the distinct kinetics of GLU-K2 kinase receptors to perform a temporal derivative calculation, extracting the rate of change in odorant concentration before the signal even leaves the bulb.
This computational precision is further refined by synaptotagmin 7, a high-affinity calcium sensor that drives asynchronous neurotransmitter release. Unlike the fast synaptotagmin 1, this isoform remains active for hundreds of milliseconds, allowing the neuron to bridge the gap between successive spikes to induce short-term facilitation. This kinetic diversity is governed by the Goldman-Hodgkin-Katz flux equation, which accounts for the extreme concentration gradients that make the Nernst equation's linear assumptions fail at the nanoscale. To manage this, the Munck's 18-1 protein acts as a molecular chaperone, clasping syntaxin 1 until the exact millisecond of vesicle priming, preventing premature snare assembly while maintaining the high-pressure readiness of the active zone. This is bolstered by calcentinine 1, an adapter that links specific cargo to kinesin motors, ensuring that high-priority receptor subunits bypass the somatic filter to reach the distal dendritic tips, effectively giving the dendrite its own localized supply chain for maintaining high-bandwidth integration.
This decentralized supply chain is the final piece of the bioenergetic puzzle, allowing the human brain to maintain a computational throughput that dwarfs our most advanced exascale clusters while consuming a mere 20 watts of power. We are witnessing a system that operates at the absolute Landauer limit of thermodynamic efficiency, where 86 billion individual processors utilize stochastic resonance to extract signal from noise.
This isn't just a biological network, it's a self-referential architecture where the laws of physics are harnessed to create a state space of connectivity so vast it exceeds the total number of atoms in the observable universe. When we look at a neuron, we aren't just looking at a cell. We are looking at the universe's most sophisticated attempt to achieve self-awareness through the cold, calculated precision of molecular machinery.